Hey guys, welcome to another video of the Usman Academy. In this video, I'm gonna tell you about the structure of the plasma membrane. In this structure of the plasma membrane topic, there are the three models that we are going to discuss. But in this video, I'm gonna discuss about the two topics, two main models. Number one model that is called the sandwich model. Second model is called the unit membrane model, and third model is the fluid mosaic model. These are the three main models, and we are going to discuss only the two models firstly. So let's start with the first model, that is called the sandwich model. According to the sandwich model that is given by the Robertson, that is given by the Danley and the Davison. Both of these scientists do the main work in 1953 on the structure of the plasma membrane, and they told us this cell that is consisting of the plasma membrane or the cell membrane, we are going to explore it. This consists of this structure, and this structure consists of the two structures in it. Number one is lipid bilayer that is present inside, that is sandwiched between the two protein layers. These are the lipid bilayer, and a layer and the two layers of the proteins are present on either sides of the lipid bilayer, and the lipid bilayer is sandwiched between the two protein layers. But here the Daniel and the Davison told us the proteins are present in them are the globular proteins. These are the type of the proteins that are very important part of the proteins, and are the globular and are the round or the globular in form. So these proteins are present on either sides of the lipid bilayer, and the structure and the thickness that is given by them is 8.0 nanometer. This is the thickness that is given by them. This structure is stabilized. How this structure is stabilized? The structure is stabilized by the attraction that is present between the protein and the lipid head. And second, this is the protein and the lipid head, and second is the tails of the lipid bilayer. Here is the attraction, and here is the traction. So these attractions stabilize the structure of the lipid bilayer, or stabilize the structure of the whole plasma membrane. And the second model that is given by the Robertson, that is called the unit membrane model. That is given by the Robertson. Robertson was a scientist, and he was also working on the plasma membrane structure, but he got all the information from the Daniel and the Davison, and he worked out in the 1960s. Instead of having the globular proteins that is present on the side of the lipid bilayer, he told that these globular proteins are not present. Instead, there are the another proteins. These proteins are called the fibrous proteins. So this is the main point that is given by the Robertson, fibrous proteins. Another thing, he told that instead of having the 8.0 nanometer thickness, this structure had the 10.0 nanometer thickness, and this structure is similarly stabilized by the attraction that is present in between them. The Robertson also told another thing, that is this plasma membrane structure is a trilaminar structure. What is meant by the trilaminar structure? On the electron micrograph, this plasma membrane is shown in the three different colors, in the three different regions. First color, that is the protein, that is shown as the dark color, and this lipid bilayer is shown as the light color, and this is shown also as dark color. So there are the three main regions: dark, light, and dark. So these three regions are known as the trilaminar structure. So this is the trilaminar structure proposed by the Robertson. There are the many similarities that is present. As you know, the Robertson got all the information from the Daniel and the Davison, so there are the many similarities that is present in between them. Some of them, the proteins are present on the sides of the lipid bilayer, and the lipid bilayer is present, or it is sandwiched, or it is present inside or in between the proteins. So, and the arrangement of the proteins and the lipid bilayer is similar in both of the models, but hence there are the many differences that is present in between them. One difference is 8.0 nanometer thickness. On the basis of the size or the thickness, there are the two differences. And second, the Robertson also told that this is the trilaminar structure, and the globular proteins here, while in them there are the fibrous proteins. So there are the tiny differences that is present between them. But both of these models, this is the thing, both of these models are not widely accepted. The reason behind them, these two models don't tell us the information about the transport of the materials, transportation of the materials, how the materials or how the water molecules transport through them. There is no information, there is no idea that is given by them. For example, the water molecules pass through them. This is the water molecules that pass through them, but they can't pass through them. This is the reason. This is the hydrophobic part. Hydrophobic part means that this is lipid region. Here we take only one. This is the hydrophobic part, this is the head region, while this is the hydrophilic. Hydrophilic means it can't allow the passage of the water molecules through it. So the water molecules stop here and they can't go further. That's the reason there is no idea that is given by them the transportation of the water molecules through the plasma membrane. And there is another thing, they can't explain the dynamic nature of the plasma membrane. Dynamic nature. In actual, the plasma membrane is very dynamic. It is not rigid, but it is firstly think that this is the rigid, but now it is thought that it is very dynamic. It's very dynamic. Hope you understand this topic, and in the next video we are going to discuss about the next model, that is called the fluid mosaic model. Hope you understand this. Thank you.